Okay, good evening to all. And today we will start GPCR in more details. In yesterday's class, we have discussed the structure of G protein coupled receptors. These are called G protein coupled receptors because they are having a common structure, means they are having N-terminal extracellular domain, and there is a C-terminal cytosolic domain. And generally, this alpha helices are passing 7 times through this plasma membrane. Therefore, they are called 7 transmembrane. Somewhere you may find that it is written like they are spanning 7 times. So 7 transmembrane. So 70M is written for these GPCR molecules actually. So these are 7 transmembrane helices. And they're called serpentine molecules. Okay, so somewhere it is written that it is serpentine. Now, okay, let me start from the beginning. So I have seen that some of the students are regular students, some of you are joining and then leaving before class and not regular. So for those students who are not attending regularly, so for 2 days actually we are discussing some of the basics of cell signaling, but as the time passes, it will become more and more complex. I'm not threatening anyone, I'm just telling the essence of this particular topic which we are going to start today. Actually, the cell signaling and signal transduction is one of the major topics from where the questions have been asked. And this need major attention to learn these pathways because these are really complex. Okay, so we will study one by one. And if today we are able to complete GPCR, then at least 60% of our syllabus, means a sense of this particular class of signal transduction is over. Okay, so because GPCR is everywhere, okay, because the GPCR are receptors which are required for smell, sight, and taste. Okay, so the receptors which are involved in our these basic things of means odorant receptors, receptors for light, vision, and the taste buds, all are signaling via these GPCR receptors. So okay, now, and be assured, because actually whatever we are discussing is everything actually is written in these PDF. This either we are using the Scooper or Pathfinder only. Okay, anything beyond this, I will give you references, I will provide you images, and the text I will share in the group only. Okay, so there is no worries, we will find related notes and supporting documents. Since everything is written in the text only, so we will see, for example, as already reviewed, most ligand responsible for cell signaling, including this neurotransmitter, peptide hormones, and growth factors, bind to the receptors on the surface of their target cell. Okay, in earlier class also I told you that majority of receptors are membrane-bound receptors, means they are present over your cell surface, your plasma membrane, and then rest are nuclear receptors. So nuclear receptor is also another class of receptors, but we are starting with membrane receptors or cell surface receptors. So now we need to understand the sense of signal transduction, how this is achieved. So when we want to study about signal transduction, we should understand one basic thing that is called amplification. So this amplification is required whenever signal bound to the cell surface receptor. Okay, so only a single molecule, or say only a single primary messenger or ligand or signal molecule can change the cell behavior totally. Okay, so actually this is not happening according to your imagination. You can think about 100-fold increase in each step when the downstream effector proteins or enzymes are activated. So we will see one by one. Okay, now, so this is the basic structure, and because these are a big family of proteins called GPCR or G protein coupled receptors, so they all have this basic structure, means you will find in each case this kind of structure. And question has been asked directly whether these domains which are passing through the plasma membrane, this is lipid bilayer, and these transmembrane helices are actually alpha helices. Okay, so if someone asks you what is the nature, whether these are alpha helices, beta pleated structures, or whatever, what are these? So these are alpha helices always. So the G protein coupled receptors, it is the largest family of cell surface receptors, transmit signals to intracellular targets via the intermediary action of guanine nucleotide binding proteins. And because these proteins are binding with GTP or GDP, depending upon whether they are active or inactive, actually when they are bound with GDP, we know GDP is less energy molecule, means GTP is a major energy providing molecule. So GTP bound form is active, GDP bound form is inactive. So these two type of things, whether these G proteins are bound with GDP or GTP, are actually working like switches, means either on or off. If they're bound with GDP, these are off. If they bound with GTP, they are on. Now, more than 1,000 such G protein coupled receptors, this type of proteins has been discovered. We know about 1,000 or more than 1,000 such proteins which are GPCR. And because these are involved in so many things, for example, smell, sight, and taste. Okay, so all this we are able to smell because of GPCR. We are able to see because of these GPCR. We are able to taste different type of items for with the help of the GPCR proteins. So actually, these are very necessary to sense the things around us. Now, actually, what is the structure? What is the basic structure? I already have told you that these are membrane spanning alpha helices, and because these are spanning 7 times the membrane, so they are called serpentine 7 alpha helices or GPCRs. The binding of ligand to the extracellular domain of these receptors. For example, there is a domain, this is end terminus of this particular GPCR protein. So this particular domain is having some of the amino acids where your ligand or a signal molecule or your primary messenger will come and bind, so that it will lead to a conformational change in this particular protein. And this conformational change will be towards this cytosolic domain side, so that some of the amino acid will come out, and so they are able to interact with some other effector proteins. For example, the G proteins which are interacting with the C domain of this particular receptor. Now, okay, one more thing is, if we talk in terms of humans, so in human genome, there are about 500 genes which are encoding for these sense of smell receptors, means these odorant receptors only. So about 500 genes in human genome which are coding for only odorant receptors, so alone. Okay, beside this we are also having sight receptors and taste receptors, so they are also GPCRs mostly. So basically, this is the largest family of proteins we are going to start today. Okay, so underline this. This is alpha helices, and then they are spanning the membrane. Now, another question which may be asked from this particular section is, what is the nature of these amino acids which are present in these alpha helices? So, because these are interacting with your lipid bilayer, and lipid bilayer is hydrophobic in nature, so obviously the hydrophobic amino acids, if you know some of the hydrophobic amino acids, these are, for example, leucine, isoleucine. Okay, so alanine, these are hydrophobic amino acids, and these are mostly present in these alpha helices, so they will able to traverse this plasma membrane. Okay. The binding of ligands to the extracellular domain leads to or induces an conformational change that allows this cytosolic domain of the receptor to bind with the G protein more efficiently, which are associated with the inner face of the plasma membrane. So in this particular diagram, it is not shown, but here somewhere in the inner leaflet of plasma membrane, there is an interacting protein called G protein. Now, Okay, so I will tell you about G protein, but before that, I want to tell you something. For example, this conformational change is written here. Actually, this is a very simple answer to many of the protein related problems, because anything in the protein is depending upon the conformational change. Okay, so this conformational change, what actually the conformational change is doing with the protein. So the conformational change will lead to the the outcome of some of the active amino acids which are making the active site of some of the domains, so that the effector proteins can more efficiently interact with these particular amino acids which are actively participating in forming this particular domain like structures. And if there is no conformational change, these particular specific amino acids or some of the portion of the protein is buried inside protein, so that the receptor is in its inactive state. So this kind of thing is a kind of auto regulation in this particular receptor or this kind of proteins, so that these can be activated in response to the conformational change in this particular protein. Now, second thing is the G protein is interacting with this particular C terminal domain of this receptor and also with the plasma membrane inner leaflet. So let's see what is this particular. Uh, okay. Now, in this particular PDF, are you able to see this GPCR? This is Pathfinder. Are you all able to assess or see this particular Pathfinder? Okay. So I think I need to share the screen each time. So this is not required when we are switching from one PDF to another, I think. Okay, fine. Now see, this is a GPCR and these are 7 transmembrane alpha helices of this particular receptor which are having 2 domains. One is called N-terminus domain where your signal molecule or the ligand molecule, or in simple term, the primary messenger molecule will come and bind. Second thing is binding of this particular signal molecule to this particular receptor, because this is very specific interaction here, leads to some conformational change. You can see that this particular C tail of C terminal domain of this particular receptor is having some of the, okay, something is coming out of this thing, means it is getting activated, or some of the amino acid which were actually buried inside in the inactive form are now coming out, so that this particular G protein, see, this is the G protein, and this particular G protein is of two type. One is monomer, one is trimeric protein. So in this case, in the most of the cases actually in signal transduction, the trimeric G protein is involved. Okay, this trimeric G protein is made up of 3 subunits. One is called alpha, beta, gamma. Beta and gamma are good friends, so they are always forming a complex. And alpha is not a good friend in the sense. Actually, when it is having no money, means when it is having GDP bound, when it is GDP bound state. So alpha is making a friendship with beta and gamma. Once GTP will come, so the GTP, because the cell cytosol, because within the cell cytosol, the concentration of GDP is less and GTP is more. So there is a chance that this GTP will replace this particular GDP from the alpha subunit. So once the alpha subunit got some GTP or energy, or you can say some of the money, it will be activated and it will forgot this particular friend of this, which is the beta gamma complex. So, and once actually this is shown not correctly in this particular PDF, the actual structure is here. So see, this is actually interacting already with this particular GPCR, but because this GPCR is inactive until and unless this particular hormone or signal molecule bound to this particular extracellular phase of this particular GPCR. Once it is bound, this particular domain will be activated. So it will be activated. So it will lead to the exchange of this alpha subunit, which is GDP bound state, is interacting with beta gamma, and actually beta gamma is also interacting with this particular receptor. Once it is activated or GTP is bound to alpha, alpha will go away and beta gamma will remain bound to this particular receptor in plasma membrane, then this alpha subunit will move and go to another protein complex which is in your plasma membrane, and this is called adenyl cyclase. This adenyl cyclase is responsible for converting the ATP into cyclic AMP. Okay, so there is another related enzyme we have discussed yesterday, but I also discussed that in today's class also. This is called guanyl cyclase, which is responsible for converting the GTP into cyclic GMP. So these are very related mechanisms and very related enzymes. But here, because we are studying about the GPCR signaling and we are going to activate the protein kinase A, so everything is related to adenine. So this ATP is getting converted into cyclic AMP so that it can activate the protein kinase A. Okay. Now, someone has query? No one. Okay, that's fine. Means you are getting the things at least. So now, where we were actually, okay. So this interaction activate the G protein, which then dissociate from the receptor, and it is talking in terms of alpha subunit only, okay, trimeric G protein, and carries the signal to the intracellular targets, which may be either an enzyme, for example, adenylate cyclase, or an ion channel. We also talk in terms of these ion channels, so I will tell you with class we will move to them. Okay. So the discovery of G protein came from the studies of an hormone, and this hormone is called epinephrine hormone. So the first hormone, which is with the help of which the GPR signaling was discovered, was epinephrine or adrenaline hormone, which is produced by your adrenaline gland or the glands which are present over kidneys. So as discussed that this particular scientist, 1970s, Martin Rodwell and his colleagues made a key observation that GTP is required to this hormonal stimulation of adenyl cyclase. उन्होंने देखा कि GTP requirement होती है so that this adenyl cyclase can be activated, and this GTP is actually required by this particular alpha subunit of this G protein. So this G protein is a trimeric G protein which is made of 3 subunits, but beta gamma complex is completely different from alpha subunit, and this alpha subunit is requiring GTP, bound GTP, which is required for its activation and dissociation from beta and gamma. Once it is GTP bound, it will go to the adenyl cyclase enzyme and will activate the adenyl cyclase enzyme, and then the adenyl cyclase will convert the ATP which is present in your cell cytosol into cyclic AMP, and then the cyclic AMP will act like secondary messenger molecule. So actually, this particular scientist, Martin Rodwell, tell us this particular alpha subunit of G protein required GTP actually. This finding led to the discovery that guanine nucleotide binding protein, that is called G protein, is an intermediary in adenyl cyclase activation. So actually, before 1970s, we do not know about this particular pathway. Okay, so it was only in 1970s by Martin Rodwell who tell us about this particular G protein, which is having a role in activation of this adenyl cyclase enzyme so that it can produce cyclic AMP. Okay, so some of the pathways are discovered in this way only. If I talk in terms of today's situation also, we will see many pathways are still not complete. So they are shown by some arrows, but actually we do not know the intermediates present, intermediate proteins which are actually doing this particular role. Okay, so we will see these things also. But for now, actually we know much about GPCR than any other signaling pathway. Okay. So now actually what is the regulatory mechanism, how these receptor molecules and how these GTP and GDP bound forms of G proteins are being regulated. So as I told you in simple words, there are three friends. One is called alpha, one is called beta, and another one is called gamma. Beta and gamma were close friends, so always making a complex, and they are not influenced by any kind of money or something. But alpha when this is having less money, for example, GDP, it will form a bond with beta and gamma. But once it got some energy, means some money, for example, GTP, it will be dissociated from the beta and gamma complex and it will move away. But it is not like that only. Once he spent this particular GTP, once the GTP is hydrolyzed and it is converted into GDP again, because we know ATP is converted into ADP, GTP is converted into GDP. So once it is hydrolyzed, it will again come back to beta and gamma and then again reassociate after this particular GTP hydrolysis. Okay, this PI inorganic phosphate will be released and the beta alpha beta gamma complexes are again formed. So that's how this particular auto regulation, means all the things is actually depending upon the GTP hydrolysis. So this tells us one thing, that कितने टाइम तक एक सिग्नल एक्टिवेट रहेगा, मींस कितने टाइम तक सिग्नल ट्रांसडक्शन होगा, ये बेटा किस पे डिपेंड कर रहा है, डिपेंड कर रहा है कि कितनी देर तक ये अल्फा फॉर्म जीटीपी के साथ में बाउंड रहेगी. Once the GTP is hydrolyzed, it will come back, and when it will come back, it will also lead to the inactivation of adenyl cyclase enzyme, because this alpha subunit, when it is activated, only able to activate the adenyl cyclase enzyme, and that's how this particular pathway, signaling pathway, is getting regulated on the first step itself by the G protein alpha subunit. Hope you are able to get this particular thing from this particular diagram. If you are not able to understand, I will repeat it again to you, but it is very simple to understand. From our daily life also we can send something, means we can understand. Okay, so most of our friends are like alpha only. So when they got some money, okay, so they will go away. Once their energy or the money is spent, they will come back to us and ask more money, ask for more money. Auto regulation. Okay, Trishla, actually so Trishla is not able to understand. I think she do not have any alpha like friends, so I need to repeat it again. So see, this is a GPCR receptor and this is a G protein. Why these are called GPCR? Because they are always complex with these kind of G proteins, which are in inactive state until and unless there is no bound hormone over the receptor. Once this hormone will come and bound, this is kind of signal molecule, will bind to the receptor specifically. It will lead to some conformational change in this particular domain, so that it will more closely associated with this particular G protein. And when this is more closely associated, means it is having high affinity, so it will also lead to some conformational changes in this particular G protein, which is made up of three components: alpha, beta, gamma subunits. These three subunits, then because we know in the cytosol there is lots of GTP. Okay, so GTP concentration is more in compared to GDP. So finally, when it push this particular subunit, it lead to some conformational change in alpha, so that the affinity for GDP will decrease and GTP will replace this particular GDP. Now this alpha subunit is bound with GTP and GDP is gone. So every time you need to remember that the fresh GTP molecule will come and replace the GDP molecule. It is not like that GDP is going to convert into GTP. It happens, it is not happening here. Okay, so every time a fresh GTP molecule will come and replace the GDP molecule. Once it is bound with GTP, now this alpha subunit will be activated. Okay, once it is activated, it will leave the beta gamma complex and will move towards the from this particular diagram it is more clear. It will move towards the adenyl cyclase complex, so that it will make an association with this particular cyclase. So this cyclase enzyme is actually responsible for converting ATP into cyclic AMP. So now first thing is this ATP is getting converted into ADP. So I also have a second screen. So this particular screen is showing that the ATP molecule is made up of three phosphates. These phosphates are alpha, beta, and gamma. Okay, so let me annotate this. So this is alpha, this is beta phosphate, and this last phosphate is gamma. Question has been asked because from this portion, so I'm telling you these things. Actually, always this gamma phosphate, okay, whenever this ATP is used by kinase, so this gamma phosphate is transferred to an substrate molecule because it is the outermost, okay, and ADP will form. And so ADP will have alpha and beta phosphate, and ATP is also having one gamma phosphate. Second thing is these bonds. So these are some high energy bonds. So they are shown like this, okay, curved lines. So these are called phosphoanhydride bonds. So these phosphoanhydride bonds are high energy bonds in this particular molecule of ATP or GTP. Okay, the only thing between ATP and GTP is if this is C double bond O and NS2 will come here, so it will be GTP. Okay, so this is adenine, and if it is replaced by guanine, then this is GTP. So in both the cases, there are high energy bonds. These are called phosphoanhydride bonds, and this particular bond is called phosphoester bond. So what is actually happening? The ATP is being converted into cyclic AMP. So we need to break this phosphoester bond. So if we break this particular bond, so you can see if you break this particular bond from, for example, here, and this particular third prime position in your sugar molecule is having an OH, free OH group. So it will attack over this particular position to make another bond. So this will lead to the formation of another molecule. Okay, so first of all, I need to remove this. Okay, then I will lead to cyclic AMP. So it will lead to the cyclization of this particular ring. So this will form a cyclic AMP. So this is cyclic AMP. Okay, now I move to, okay, so now so this ATP is getting converted into cyclic AMP. Now suppose this alpha subunit, which is bound to GTP in its active form, utilize its GTP and hydrolyze GTP into GDP, then it will again inactivated and will interact again with the beta gamma complex. So that is more clear from this particular diagram. So in the GTP bound state, it is active and activating your adenylate cyclase enzyme. But once this GTP is hydrolyzed, so inorganic phosphate will be released, which is actually the gamma phosphate. Okay, so this is again and again having this bound GDP, and in this particular diagram, it will again interact with the beta gamma complex. Okay, who is this? Trishla, please rejoin if you are facing some problem. Okay. Now, so we have discussed everything from this particular section. Now, the next thing I want to tell you is, okay, so this trimeric G proteins, alpha, beta, and gamma in mammals actually, in case of humans, for example, in mammals, these are encoding 20 different alpha subunits, 5 beta subunits, and 12 gamma subunits. So you can suppose that this kind of diversity is present within our genome. Our genome can encode for 20 different alpha subunits, which are able to bound to GDP or GTP, and 5 beta subunit and 12 gamma subunit. So they can form complexes with one another, and therefore they can be classified into 4 different families, which are given in your Pathfinder. Okay. Uh, I hope most of you are having the Pathfinder, and actually I'm having 7th edition. So I'm telling you page number so that you can find these classification. In my book, this is 356, where they have classified these G protein based on these alpha, beta, gamma subunits into uh, okay, G alpha S means S for stimulatory, G alpha I for inhibitory, or 0 for neutral, G alpha Q11 and G alpha 12/13. So actually this is given. If you do not having this particular classification in your books, I will share in the group after the class. So please focus on class. If you do not have, no worries. Okay, I will share, nothing to worry about. So now, okay, so after this, actually the receptor tyrosine kinase is given in this particular book, Cooper. So I will not, I will come to this particular section after completing GPCR. Otherwise, everything will be mixed up and, oh my God, mixwise will be prepared. So I'm skipping all this section. So I will come back and I will tell you after we complete our GPCRs. So now again, after some pages or 2 or 3, again the GPCR signaling is given in your book, Cooper. So you can find it from here. The page number, I don't know, this is 641 actually, according to this. And if from the book, I tell you it is some 617, okay, exactly. Now, what is the pathways of intracellular signal transduction? We have seen that, okay, it's time to explain it from here actually. So we have seen that there is a GPCR which is present in your plasma membrane. It is interacting or not interacting with some of the G protein. Actually, it is interacting better with this particular G protein when it is bound with some signal molecule, because it leads to some conformational change in its C-terminal domain or C terminus. Okay. But in this book, what is the problem actually here is this beta and gamma complex is shown on this side, whereas this is generally present on the side of the receptor actually, because it is interacting with the C-terminal domain, and alpha domain is generally present, but there is no problem. Everything is fine. Okay, so this alpha, beta, gamma is clearly visible in this particular section. So this is your G protein, this is your GPCR. So once GTP will come and replace the GDP from the alpha subunit, it will lead to the activation of alpha subunit, and then your alpha subunit will have less affinity for this beta gamma complex. Now, actually, we need to understand now actually what will happen inside the cell once this particular signal will bound outside the cell. And this is actually the real signal transduction, because the signal will not going inside the cell and it is sending its masses across the plasma membrane. That is the real essence of signal transduction. Okay, so I'm coming to Cooper again. Now it is much easier to explain. Okay, so many cell surface receptor, for example, we have seen GPCR. And second thing is receptor tyrosine kinases, or these are also the surface receptors. So they will explain everything before this particular section. So many of the cell surface receptors, I'm giving reference of GPCR only here because I do not discuss with you about receptor tyrosine kinases. So for now, you can understand how these GPCR are present over cell surface and how they are interacting with different signal molecules, for example, hormone epinephrine. Stimulate intracellular target enzymes, which may be either directly linked to or indirectly coupled to the receptors by G proteins. So actually these G proteins are important because these are taking part in the activation of cyclic AMP formation, okay, by the activation of adenyl cyclase enzyme. These intracellular enzymes serve as downstream signaling elements that propagate and amplify the signal initiated by ligand binding. तो देखो सिग्नल ट्रांसडक्शन कब होगा जब एक ligand molecule, एक proper signal molecule will come and bind to these particular surface receptors. In most of the cases, a chain of reactions transmits the signal from the cell surface to a variety of intracellular targets. For example, this is called intracellular signal transduction. Actually, if someone asks you what is intracellular signal transduction, so simply you can explain that if a signal molecule will come and bind to the cell surface receptor, so the signal, without being going inside the cytosol, is able to transmit the signal inside the cell. And this is actually happening with the help of several chains of reactions which are transmitting your signal inside your cell cytosol. And actually, one molecule is able to transmit this particular signal because there is something called amplification. So the target of such signaling pathway frequently include transcription factors that regulate the gene expression. So these transcription factors are involved. I will discuss one of the transcription factor, for example, SCRAB, cyclic AMP response element binding protein, that is called CRAB, is one of such transcription factor we are going to discuss in our next page. Okay, so frequently transcription factors which are able to regulate the gene expression, and how the gene expression is going to affect our metabolic pathways, we will also discuss with the help of these epinephrine regulated metabolic pathway, means the glucose breakdown actually. Intracellular signaling pathway is thus connected to the cell surface with the help of these signal transduction or intracellular signal transduction. So leading to the changes in gene expression in response to extracellular stimulus or hormone binding. Now, because something is given in this particular diagram is, I already have told you that this ATP is converted into cyclic AMP by an enzyme called adenyl cyclase, which is activated by GTP bound form of G protein, alpha subunit. So you know this. Then what is the second level of regulation? I told you that this alpha protein or the alpha subunit of G protein is being regulated by bound GDP and GTP and acting as a switch. Means if it is bound with GDP, it is an inactive state. If it is bound with GTP, because with GTP is high energy molecule, so it will be activated. Similarly, the second step of regulation is either to make the cyclic AMP, that is a secondary messenger, or to break down the particular cyclic AMP if it is not required further. So who is going to degrade this particular cyclic AMP? So cyclic AMP is produced in large amounts because it is a secondary messenger and it will go here and there within the cell cytosol and activate some of the target enzymes, and one target enzyme is protein kinase A. So this is protein kinase A actually. It is made up of 4 subunits. So this is a protein made up of 4 subunits, and sorry, something has happened and my screen is, oh, Arti is not getting my sound. So Arti, is it clear now or still you're facing the same problem? So apologize because something has happened due to which my screen is minimized somehow. I again shared my screen and I want to know whether you are able to see the screen and my voice is not clear to Arti. So Arti, please answer whether you are able to hear me now or. So guys, sorry, and actually something has happened. So my network problem actually, I have restarted my router. So hope this will work better now. Okay, Aarti. So actually, I'm starting from here again. So actually, I was here. So we have discussed that actually the second switch is this cyclic AMP, because we know cyclic AMP is as secondary messenger within our cells. It is going to activate this particular enzyme, protein kinase A. So this should be required for further signaling. So the second step of regulation is either to synthesize this particular cyclic AMP if it is required, and if it is not required within the cell, so it can be broken down. Means the cyclic AMP phosphodiesterase is going to break this particular phosphodiester bond and restoring your, yeah, this is not cyclic, so this is simply called AMP. Okay, so now intracellular signaling was first elucidated by studies of action of hormone, this particular epinephrine hormone we have discussed. So epinephrine, or this adrenaline hormone, is going to transduce the signal via GPCR. So suppose this GPCR means this epinephrine receptor is also a GPCR, so it will bind with epinephrine. So it is responsible for the signal which is leading to the breakdown of glycogen to make glucose in the anticipation of muscular activity. So whenever you do physical exercise, you will require lots of glucose. So from where you're going to get the energies? From the stored glycogen in your muscles. So this glycogen in your muscles, or muscle glycogen, should be broken down, and this broken down is actually done with the help of this particular GPCR pathway. So now, so up to here it is clear to you, and now I will move to Pathfinder so that we can move further. So it is clear. The epinephrine will come and bound to your GPCR. GPCR conformational change will lead to the activation of this alpha subunit, and this alpha subunit, when bound with GTP, and it will go to adenyl cyclase enzyme, which is an effector enzyme of this pathway. So it will produce lots of cyclic AMP. The cyclic AMP will not be degraded at this step. It will only degraded once the signal is over, okay? Or the cyclic AMP is not required any further. So this cyclic AMP now is going to directly activate cyclic AMP dependent protein kinase. So therefore, because it is cyclic AMP dependent, so this is protein kinase A for AMP dependent. So cyclic AMP also induces opening of certain type of ion channels in plasma membrane of some of the highly specialized cells, for example, heart cells. Protein kinase A catalyzes the transfer of terminal phosphate group from ATP. The terminal phosphate group is gamma phosphate actually, to a specific serine or threonine of the selected target protein. So the question may be asked, so what is the nature of this protein kinase A? This is a serine or threonine kinase. So you need to remember it. If someone asks you what is the nature of this particular protein kinase A, so this is either you say this is a serine or it is a threonine kinase, means threonine kinase. So it is going to phosphorylate either serine or threonine, not the tyrosine, not any other amino acid. So remember, uh, because they are evolved in such a way that they are specific for some of the amino acids. So this protein kinase is a serine threonine kinase. Similarly, many enzymes will come, and I will tell you at each step whether it is going to phosphorylate serine, whether it is going to phosphorylate threonine, or whether it is going to phosphorylate tyrosine. Some of the enzymes then evolved in such a way that they are able to phosphorylate either serine or threonine, but not tyrosine. Some of the enzymes are also known which are able to phosphorylate threonine and tyrosine, but not serine. Okay, so this may happen. Therefore, this has become very important and urgent to remember what is the nature of this proteins or enzymes what is the target amino acids for these phosphorylations? Because we know these are kinases, and kinases do their function of phosphorylating some of the target proteins. Okay, so now this protein kinase A, which is having some serine and threonine residues in its amino acid sequence, is made up of two catalytic subunit and two regulatory subunits. So it will be phosphorylated over serine or threonine residues. So C. And once it is phosphorylated, it will be activated. Now I will tell you how it is going to be activated. So it is actually given in both the books. In your Pathfinder also, it is very clear, and in your Cooper also it is very clearly given that. Okay, so these subunits, these regulatory subunits are large, catalytic subunits are small, but it is made up of four subunits. So the phosphorylation will actually happen. Oh, okay. So, sorry, it will phosphorylate some target proteins, not this particular protein phosphorylated. So, it will be activated with the help of cyclic AMP, which is being produced as a secondary messenger. Okay, so the cyclic AMP will come and it will have a four binding sites over two regulatory subunits. So, each regulatory subunit bound with two cyclic AMP molecules, so that the conformational change will happen with the regulatory subunits and the affinity for catalytic subunit will be reduced, so that catalytic subunit will be dissociated from the regulatory subunits. Now, question is, after dissociation, what will happen? So it will be activated. So this protein kinase A catalytic subunit will be dissociated, and now this is called activated protein kinase A. Now this activated protein kinase A catalytic subunit will go inside your nucleus. So this is nucleus of the cell, and this is nuclear pore complex. Through nuclear pore complex, these catalytic subunits, because they are going to catalyze some of the phosphorylation events over serine or threonine, they will move inside your nucleus and they will phosphorylate the crab protein, cyclic AMP response element binding protein. Okay. So once the crab is phosphorylated over specific serine threonine residues, it will be activated. So once it is activated, it will act as a transcription factor and stimulate the gene transcription of some of the target genes. See how. So see from here, actually, where it was. So next page actually. So see, this is the regulatory subunit, this is the catalytic subunit, and complete. This is called protein kinase A. This protein kinase A, because it is going to activate in response of cyclic AMP, the cyclic AMP will come and bind to its respective position, so that the affinity of catalytic subunit will decrease for regulatory subunits. And this catalytic subunits, after getting free in the cytosol, will move through the nuclear pore complex into the site in the nucleus, nucleoplasm. Okay, so in the nucleus, then they will phosphorylate this crab protein. So these crab are actually transcription factors, and they will bind over this particular response element. I also told you that what is the response element? A sequence of DNA over which this particular protein is going to bind specifically is called uh, crab CRE, regulatory element. And this is called SCRAB, cyclic AMP regulated response. Okay, so response element binding protein. So now this catalytic subunit will add one of the phosphate over the serine or threonine residue in this particular protein from ATP only. So ATP is converted into ADP, and gamma phosphate is being transferred to this CRAB subunit. This CRAB is forming a heterodimer, and a co-activator will come and bind to this particular complex, so that the transcription of the target gene will be activated. Now the question is, what gene is under direct control or direct regulation of this particular pathway? So actually we are looking after epinephrine GPCR signaling. So this epinephrine gene is having some of the genes under its control. For example, it is given here. Okay, so see, this is the protein kinase activated form. This is catalytic subunit actually. So this catalytic subunit is going to phosphorylate your respective proteins. So one thing, it is phosphorylated your trap protein in the nucleus. In the cytosol also it is going to phosphorylate some of the proteins. For example, it is going to phosphorylate phosphorylase kinase, glycogen synthase, and glycogen phosphorylase. Suppose now how it is going to regulate the metabolism. Now question is how it is regulating our glucose synthesis from the glycogen in the muscles when we are doing the physical exercise. So this protein kinase A, first thing, it will phosphorylate every protein it will find. So it will phosphorylate the two proteins. One is phosphorylase kinase, which will be activated after phosphorylation. On the other hand, it will inactivate the glycogen synthase because the glycogen synthesis is not required. We are doing some physical exercise, so we need to break the glycogen, not going to synthesize it. So this should be inactivated. So this phosphorylation will going to inhibit this particular enzyme. So once the glycogen synthase is phosphorylated by this particular protein kinase A, it will be inactivated. Now the phosphorylase kinase is also a kinase. So once it is phosphorylated, it will be activated, and it is going, because it is a kinase, it will going to phosphorylate some other proteins. So next step is to phosphorylate glycogen phosphorylase. Now this protein kinase is not having any role. Once the phosphorylase kinase is activated, now this phosphorylase kinase, when activated by phosphorylation, is going to phosphorylate the glycogen phosphorylase. When this glycogen phosphorylase is activated, means phosphorylated, then glycogen phosphorylase will also become activated, and it is going to break your glycogen into glucose 1-phosphate. Now, the question of this year gate only. So it was asked that the when the glucose is undergoing glycolysis, it is producing 2 ATP. But when it is formed from glycogen, because the glucose 1-phosphate is produced, so how many ATP will be produced? Now this time the 3 ATP molecules will be produced. So every time when glucose 1-phosphate is broken down by glycolysis, so it is undergoing glycolysis, so it will lead to the synthesis of more number of ATPs, and therefore we are able to do the physical exercise. Okay, I hope this is clear, and I have complete this particular exercise. Now you are able to understand how your metabolism is going to affect by GPCR signaling at least. So it is going to enter into the glycolysis pathway, and the ATP molecules are generated, which are the energy currency for our cells. So that's how our cells are able to regulate the ATP production in response to GPCR signaling. So I'm giving you 5-minute break now so that you can analyze the things which we have discussed. And if you're having some query up to here, you may ask. Okay, you are free to ask. So Shreya is asking, epinephrine is the intracellular hormone. Yes, beta, it is a hormone, and it is also a signal molecule which is working by GPCR signaling. So we have seen this we discussed. Ankita is asking what is the difference between protein kinase and protein kinase C. Okay, so actually this protein kinase A is under the regulation of cyclic AMP. So because it is AMP, so it is A's name is like this only. Protein kinase C is under the regulation of calcium ions, so it is the C is taken from there on. So I will tell you about this calcium regulation also, but not now. Okay, so once we finish this exercise, then we will move to protein kinase C. So now protein kinase A is clear or not? I'm asking this only. Okay, fine. So everything is written in the text, and if you go through the text, you will get everything. And actually, the second portion which we are going to discuss in today's class is cyclic AMP. थोड़ा सा मैंने कल भी इसको डिस्कस करा था, but today also I will tell you more about the cyclic AMP. So cyclic AMP and cyclic GMP both are secondary messengers. Okay. So जैसे cyclic AMP प्रोड्यूस होता है और रेगुलेट करता है, वैसे ही cyclic GMP भी रेगुलेशन में अपना रोल प्ले करता है, लेकिन some other GPCRs, not in case of epinephrine actually. तो अभी हमने डिस्कस करा कि कैसे एपिनेफ्रिन मेटाबॉलिज्म को अफेक्ट करता है, जब हम फिजिकल एक्सरसाइज करते हैं, भागते हैं, दौड़ते हैं या फिजिकल वर्क करते हैं, फॉर एग्जाम्पल वी रिक्वायर लॉट्स ऑफ एनर्जी. So our muscle glycogen stores will be broken down into glucose, and this glucose is actually glucose 1 phosphate, which is when entering into glycolysis providing more energy. This question has been asked in 2020 gate only, and in many examination also previously it is being asked. So you need to remember the things from the class only, so these things are generally asked, so these are important. Now, similarly, the cyclic GMP is also an important secondary messenger in animal cells, but uh, not in the epinephrine pathway. It is playing a role in your vision, means you are able to see in the dim light or in the presence. So in our eye, it is having very important role. Important second messenger in animal cells, although its role are not as clearly understood as that of cyclic AMP. बिकॉज इतना ज्यादा रिसर्च इस पे नहीं हुआ है, और इसलिए जितना हम साइक्लिक एमपी के बारे में जानते हैं उतना शायद साइक्लिक जीएमपी के बारे में हमें नहीं पता है. वी डू नॉट क्लियरली अंडरस्टूड हाउ इट एक्चुअली वर्किंग, बट एक्चुअली वट एवर वी नो वी विल डिस्कस इन दिस पार्टिकुलर सिग्नल ट्रांसडक्शन क्लासेस. So cyclic GMP is formed from GTP. Similarly, जैसे कि cyclic AMP ATP से बन रहा था, वैसे ही cyclic GMP बनेगा GTP से. लेकिन यहाँ पे जो एंजाइम होगा वो होगा guanyl cyclase instead of adenyl cyclase enzyme, and degraded to GMP by the same enzyme that is called phosphodiesterase. Okay, so this phosphodiesterase is going to break down. So this is cyclic GMP specific, which is going to degrade it into GMP. So it is when it is not required, it will be broken down. Now, yesterday we have discussed about nitric oxide and carbon monoxide signaling, और थोड़ा सा मैंने बताया था कि कैसे वो हमारी आई के रेटाइना में और रॉड सेल्स में एक्चुअली रोडॉप्सिन जीपीसीआर की हेल्प से हमें देखने में हेल्प करता है, हमारे विजन में हेल्प करता है. So as discussed earlier in this chapter, guanyl cyclase are activated by these guanyl cyclase. This particular cyclase is going to convert your GTP into cyclic GMP, which is the secondary messenger, and this particular guanyl cyclase is activated by nitric oxide and carbon monoxide, as well as some peptide ligand molecules. Now, stimulation of these guanyl cyclases lead to the elevated levels of cyclic GMP, which is a secondary messenger, which then mediate the biological responses. So nitric oxide का best response क्या था? It is going to dilate your blood vessels so that the blood flow will be increased. Okay, and your muscle relaxation में role play कर रहा था. So the action of cyclic GMP is frequently mediated by the activation of cyclic GMP dependent protein kinases, although cyclic GMPs also regulate ion channels and phosphodiesterases. So in this example, in our eye, this cyclic GMP is able to regulate the ion channels. And क्या होता है, hyperpolarization हो जाता था, और hyperpolarization की वजह से क्या होता था, हमारा जो sodium ion channels हैं वो close हो जाते थे, है ना. So we have discussed it yesterday also. Today also we will discuss it into great details. So, and the phosphodiesterase enzyme, because this phosphodiesterase is going to degrade your cyclic GMP when it is not required, it will be broken down so that this phosphodiesterase is degrading your cyclic GMP. Cyclic GMP concentration in the cytosol will decrease, and thus the signal transduction will happen. यहाँ पे उल्टा है, जैसा कि हमने अभी पढ़ा कि साइक्लिक एएमपी की कंसंट्रेशन बढ़ती है, वो प्रोटीन काइनेज ए को जाके एक्टिवेट करता है, और फिर प्रोटीन काइनेज ए अलग अलग प्रोटीन्स को फॉस्फोराइलेट करता है या ट्रांसक्रिप्शन फैक्टर्स को फॉस्फोराइलेट करता है न्यूक्लियस में. यहाँ पे साइक्लिक जीएमपी का रोल जो विजन में है, जो आई में है, it is completely different. The concentration is decreasing by this phosphodiesterase enzyme, and this decreased cyclic GMP is going to influence the ion channels, associated ion channels, so that you are able to see the things. Okay. Now, what is actually happening here? Okay. So now see here. Okay, so one well-characterized role of cyclic GMP in vertebrate eye, where it was serving as a secondary messenger responsible for converting the visual signaling and received as light to nerve impulses. So it is going to convert your light signal into nerve impulse so that your brain can study these nerve impulses, and you are able to analyze the things which you are able to see. Okay, so the photoreceptors on the rod cells in the retina is actually GPCRs, and these GPCR proteins are called rhodopsin proteins. So rhodopsin is activated as a result of absorption of light by associated small molecule that is called 11-cis retinal. And this molecule is covalently attached with this particular protein that is called opsin protein in this particular rhodopsin molecule. So, which then isomerizes to all transretinal forms. So, जब light आएगी, when the light signal come, it will convert the 11-cis retinal into all transform, and this all transform is going to do some conformational change in GPCR rhodopsin, so that the G protein can be activated. Okay, so we will see one by one, step by step. But before moving to that portion, actually, we have discussed protein kinase A, and I tell you at every step of regulation. तो पहला जो step था उसमें क्या हो रहा था, cyclic, oh sorry, this G protein alpha subunit is bound with either GDP or GTP. When it is bound with GTP it is activated, when it is bound with GDP it is inactivated. तो पहला रेगुलेशन तो उस स्टेप पे हो गया. दूसरा स्टेप पे रेगुलेशन कहाँ हुआ, जब हमारा साइक्लिक एएमपी या साइक्लिक जीएमपी बन गया, साइक्लेज एंजाइम ने बना दिया, either adenyl cyclase or guanyl cyclase, so it will be degraded by phosphodiesterase enzyme. So the second step of regulation is to regulate the concentration of secondary messenger molecules, whether it is required or not required, depending on that particular situation. Either it is synthesized by cyclase enzyme or it will be degraded by phosphatase enzymes. Now, the third step of regulation is to do the reverse of protein kinase A. So, once this cyclic AMP will be produced, it will activate your this particular enzyme that is called protein kinase A. Catalytic subunit will go inside the nucleus or phosphorylate some of the protein in the cytosol. For example, we have seen glycogen synthase, glycogen phosphorylase, and so on and so forth. But the thing is, to reverse the things means the protein which are giving phosphorylated, if they are remain in phosphorylated state, so they will continue to do their job. So after the physical exercise is over, they will continue to degrade your glycogen stores and they will deplete all the stores. So it is required when the physical exercise is over, the glycogen should be synthesized again so that it can be required next morning. So okay, so these phosphorylated amino acids, phosphorylated serine or threonine residues should be removed. So these phosphorylations are removed by one enzyme that is called as phosphatase enzyme. Actually, protein phosphatase 1. So this protein phosphatase 1 enzyme is able to remove this particular phosphorylations which are done by protein kinase A. So this is third step of regulation where the protein kinase A can be reverted back, the function or the action can be reverted back by protein phosphatase. So kinase and phosphatase are having opposite functions or roles, therefore they can control each other's activity. तो एक balance बना रहेगा, जितने phosphorylations होंगे तो प्रोटीन फॉस्फेटेज A1 और A आएगा और उन फॉस्फोराइलेशंस को रिमूव कर देगा so that they can be inactivated or activated. Not always inactivated, because we know glycogen synthase will be activated once it is the phosphate group is removed, so that the glycogen synthesis can happen. So see here, it is inactive in phosphorylated state. So glycogen synthase will be activated once this protein kinase, protein phosphatase 1 will remove this phosphate. So this will be activated. So glycogen synthesis will be activated again. On the other hand, protein phosphatase 1 remove all these phosphates so that they can be restored in their inactive form. So that the cell can maintain or balance its synthesis and degradation of glycogen. Okay, so I hope this is clear to each student so that I can move to the next discussion. Fine. So we have seen this, we have seen about in the eye, it is also having some role. Okay, so we need to discuss this particular section in our eye retina. This particular rhodopsin in our rod cells, we are having two types of cells. One is called rod cells for the dim light vision or black and white vision, and another type cells are called cone cells, which are required for colored vision. So see here, this retinal is a molecule which is associated with this rhodopsin, and it is making some covalent bond. Okay, so if it is not clear from here, I will move you from here to some other PDF from where you can get it better. So move here, here, here. Okay. Okay, these things also we need to discuss, but okay, first let me finish this and then I will move to the some pages I have left in Pathfinder, so I will clear all your queries from those sections. So see, this is 11 cis retinal, which is a molecule shown here, this yellow colored molecule here. This is 11 cis retinal. Okay, and it is bound with this particular rhodopsin protein. So see here it is shown something like this. So, okay, so this is opsin. What is opsin? Opsin is the protein component of this rhodopsin. Rhodopsin is a GPCR and it is having some protein component that is called opsin. Opsin is covalently linked to your 11 cis retinal. So this 11 cis retinal is coming from vitamin A or beta carotene, which we take in our diet. So now you can understand this vitamin A or beta carotene is having very important role in our diet. So we should take it in our in diet on regular basis. So okay, so when light will come, when a light signal will come, or this, so it can be seen from here also. When this light signal will come, so see here, this light is coming and it will doing some change in this particular retinal component. So this covalent linked retinal. So see here, this 11 position, it is cis because both the bonds are towards the same side of this double bond. So this is called cis retinal and it will be converted into trans form. So this is a double bond and we can see the two bonds are on opposite side. One is moving up and one is moving down. So this is called trans. So in this particular diagram, you can see whenever there is double bond, so all trans are present. So this is trans, this is trans, this is getting converted into trans in response to light signal, and this is called all transform. So always remember, whenever there is a light, this 11 cis form will be converted into all transform, and this all transform is going to do some conformational change in opsin because it is moving now. So it will, because this protein is also covalently linked to this particular molecule. So it will also move some of the amino acid in this particular opsin component, protein component, so that conformation of the protein will change. And this conformational change is responsible for activation of this particular GPCR. See, vision depends on this particular GPCR that regulates cyclic nucleotide gated ion channels. So as I told you that in some cases the GPCR are controlling these ion channels. So the first step in vision is the absorption of light energy by this particular rhodopsin protein. Okay, so the rhodopsin is the integral protein with 7 transmembrane spanning helices or helices present in our rod cells. Underline this, rod cells. Light absorbing pigment that is called chromophore is actually this 11 cis retinal. So okay, so this is important. This is chromophore, colored pigment. Okay, is covalently attached to our opsin, protein component of rhodopsin. This opsin is a protein component of our rhodopsin, which is a GPCR. When the photon is, because we have seen the, okay, so some of you may have doubt in your head why this protein component, because generally we know these GPCRs are proteins. Not true always, because in our first diagram also we have seen, okay, let me show you something. So here, okay, here. So see here, the GPCRs are proteins and some carbohydrate molecules are also bound to the extracellular surface. So they are glycoproteins actually. Okay. So GPCR are glycoproteins. So the protein component is called opsin and rhodopsin. Now it is more clear, I think. Someone is having a query. So, okay, Shreya, actually this protein kinase 1 and protein kinase, what is PKA? Yes, yes, yes. Protein kinase A and protein phosphate. This is protein phosphatase 1 beta. This is PP1, protein phosphatase 1. And protein kinase A will work in opposite fashion to each other because one is kinase, one is phosphatase. And Shreya, you know, the kinase is going to phosphorylate the protein, whereas phosphatase is going to remove the phosphate group from the protein. So I'm coming to your topic. Okay, I will tell about these enzymes more. So it is given in your Pathfinder also. So I will lead you to that particular topic. Let me finish this particular section because this is very important and the many questions can be asked from this particular section. So please focus here, all of you. Now this light absorbing pigment, that is chromophore 11 cis retinal, is covalently attached to this particular opsin protein, which is a protein component of the rhodopsin actually, because rhodopsin is a glycoprotein. When the photon is absorbed by the retinal component of rhodopsin, the energy causes a photochemical change and it will convert this form, cis form, into all transform. So in the excited conformation, when it is excited, so rhodopsin interact with the second protein that is called transducin, which is very close to this particular protein. So, okay. So if it is not clear from here, I will lead you again to this particular book. Oh, sorry. ज़्यादा ही चले गए. Sorry. अभी ये सब मत देखो, otherwise you will be confused. We will come to them one by one. Okay, so there is another protein, G protein, that is called transducin protein. So it is going to interact with this particular protein. When it is interacting with this particular protein, so it will be activated because this alpha 7 will bond with GTP, and once bound with GTP, it will go to phosphodiesterase enzyme. See, this is the difference here. When it is bound, when we are talking in terms of GPCR epinephrine signaling, this alpha subunit, when bound to GTP, is going to activate adenylate cyclase enzyme so that this cyclic AMP is produced. But here in our eyes, it is having some different effector target proteins. So here it is going to activate phosphodiesterase directly so that it can degrade the cyclic GMP directly. Okay, so this is phosphodiesterase, which is specific for cyclic GMP. So cyclic GMP will be degraded and this phosphodiester bond will be degraded, so the GMP will be produced. And GMP is not a secondary messenger, only this is a secondary messenger. So the cyclic GMP or secondary messenger is depleted from your cell cytosol or rod cells so that signal can be transduced. Okay, so there is many ways to transmit a signal, many ways to for signal transduction actually happening. Okay, so if you are able to get this, so this transducin will go and directly activate the phosphodiesterase enzyme so that cyclic GMP will be converted into GMP. So this cyclic GMP is opening your ion channels, sodium ion channels, but once it is inactivated, so it will not able to, okay, so it will not able to open these sodium ion channels. These will be closed and therefore there is no influx of sodium ions within your cell, so that there is a hyperpolarization state will be achieved. See, so in the dark, GDP is bound with this transducin protein, but once the light will come and everything is changed, so so when there's light, the signal will be sent. It interacts with the transducin, catalyzing the replacement of bound GDP with GTP from the cytosol. You are able to understand, but I'm telling because I have given so much time in discussing all these things only. So So okay, so what will happen at the end? The cyclic GMP concentration levels will fall in the cytosol. The concentration will decrease. So this drop in the cyclic GMP concentration lead to the decrease in amount of cyclic GMP, which is bound to plasma membrane sodium ion channels, allowing more of these highly cyclic AMP sensitive channels to close. जिसकी वजह से क्या होगा? ये चैनल बंद हो जाएंगे भैया, और बंद होने के बाद में कोई भी सोडियम आयन अंदर नहीं आएंगे. So closure of these channels inhibit the influx of sodium ions. So these channels are kept open in the dark क्योंकि dark में कोई भी signal transduction नहीं होता. There is no signal sent in the dark. Okay, so everything is happening because there is light. So closure of these animal okay, so it is done. In this way, light causes a hyperpolarization rather than depolarization of the plasma membrane. We have discussed this section yesterday. Depolarization happens first when these sodium ion channels are closed, and then the potassium ion channels will open, and then there is repolarization. Repolarization is followed by hyperpolarization. Okay, so this hyperpolarization is happening here in this particular case. So this is very important. A number of mechanisms operate rod cell to allow the cells to revert quickly to the dark state. So first thing is sodium ion channel closes in response to light are also permeable to calcium ions. So अब देखो इनका रेगुलेशन कैसे होता है. वैसे तो क्या होगा, साइक्लिक जीएमपी अगर डिग्रेड हो गया तो ये आयन चैनल क्लोज हो जाएंगे, लेकिन एक्चुअली जो ये सोडियम आयन चैनल्स होते हैं कैल्शियम आयंस के लिए भी परमेबल होते हैं. ओके, in your rod cells these calcium ions are also can enter through these sodium ion channels. So once the calcium will move through these channels, we know calcium is having more positive charge, two positive charge in contrast to sodium ions. So that when they close, the normal influx of calcium ions are also inhibited, causing the calcium ion concentration in the cytosol to fall. The decrease in calcium ion concentration stimulate guanylyl cyclase to replenish the cyclic GMP. So this guanylyl cyclase enzyme in the rod cells are under direct control of calcium actually. So once this calcium concentration will be low, it will activate these guanylyl cyclase. So these guanylyl cyclase enzyme will produce more cyclic GMP, and the cyclic GMP will be produced. It will bind to these channels so that it will allow again the movement of sodium and calcium ion channel in the cytosol. So that's how these are being regulated, so that rapidly returning to the level where it was actually in the dark actually. Okay, so a specific calcium ion sensitive protein that mediates the activation of guanylyl cyclase in response to fall in calcium ion levels in cytosol. This protein is actually the rhodopsin specific kinase or serine kinase, which phosphorylate the, oh sorry, this is second type of regulation. So you got this particular thing in contrast to cadmiol. Okay, so there is a protein in our muscles that is called calmodulin protein. So we know calmodulin is activated when calcium is bound to this particular protein, but actually जो protein इसको regulate कर रही है eye में, retina में, rod cells में, it is not regulated by calcium ions. Actually, it will be active when it is calcium ion free. So when calcium ion will bind to this particular protein, it will be inhibited in a very opposite fashion as calmodulin function. Okay. Second type of regulation in the retina or the rod cell is the rhodopsin specific kinases, which is a specific serine kinase. Now, actually, I told you this thing also. You need to note these things because in retina, this particular rhodopsin specific kinase is a serine kinase. It is not going to phosphorylate your threonine or tyrosine. It will specifically phosphorylate only the serine residues. Cytosolic tail of activated rhodopsin GPCR and partially inhibit the ability of rhodopsin to activate the transducin. अगर बेटा ये इसका फॉस्फोराइलेशन कर देगा तो उससे क्या हो जाएगा कि जो तुम्हारा एक्टिवेटेड rhodopsin है वो ट्रांसड्यूसन के साथ एक्टिव उसको इंटरेक्ट नहीं कर पाएगा और उसको एक्टिवेट नहीं कर पाएगा. पार्शियली जो है वो इनएक्टिव हो जाएगा, पार्शियली इनएक्टिव. अब पूरा इनएक्टिव कब होगा? I will tell you. So an inhibitory protein that is called arrestin protein is also having some role in retina rod cells. So these bind to the phosphorylated rhodopsin C terminus. So further binding, further inhibiting this particular rhodopsin activity. So पूरा inhibit कब होगा? जब एक arrestin protein आके इस phosphorylated serine के साथ में bind हो जाएगी on the C terminus of this particular rhodopsin or GPCR. So at the same time, the rhodopsin is being shut off, regulator of G protein signaling protein or GS protein. There is another protein called a regulator of G protein signaling. So it will control the G protein. Okay, so which act like GTP is activating protein. क्योंकि ये क्या करेगी? GTP को एक्टिवेट करने का काम करेगी. वैसे तो हमें पता है कि एक इनबिल्ट सब यूनिट होती है क्योंकि जो जी प्रोटीन है इट इज ट्राइमेरिक प्रोटीन एंड अल्फा सब यूनिट इज GTPase function. Alpha subunit is having inbuilt mechanism to degrade to hydrolyze the bond GTP. But there is another protein reported that is called RGS, regulator of G protein signaling, which act like to GTPase activating protein, जो alpha subunit की GTPase activity को further enhance करती है, so that जी प्रोटीन की अल्फा सब यूनिट के साथ में जो बाउंड जीटीपी है उसको जल्दी से जल्दी हाइड्रोलाइज करके जीडीपी में कन्वर्ट कर दिया जाए, और जब वो जीडीपी में कन्वर्ट हो जाएगा तो वो वापस से जाके बीटा एंड गामा के साथ कॉम्प्लेक्स बना के फॉर्मेशन करके इनएक्टिव हो जाएगा. ओके, so that what will happen in this case, so not from here actually, so what will happen, so once this particular GTPase, alpha subunit is a GTPase, So GTP is hydrolyzed. The GDP will form a complex of beta and gamma. So it will not able to activate phosphodiesterase enzyme, so that the cyclic GMP will not be further degraded into GMP form. और इस तरह से क्या होगा, जो हमारे चैनल्स हैं, साइक्लिक जीएमपी की कंसंट्रेशन धीरे धीरे बढ़ने लगेगी, ओके, और फर्दर कम नहीं होगी, सो दैट द चैनल्स कैन बी ऑपरेटेड, सोडियम एंड कैल्शियम आयन चैनल्स, rod cells. Now this is flow chart is showing very clearly that light will come, conformation of rhodopsin will change in the response because 11 cis retinal will be converted into all transform. It will lead to the activation of some protein, G protein, that is called transduction. Phosphodiesterase will be activated so that the cyclic GMP will be degraded, decreased concentration intracellular cyclic GMP. A closure of sodium ion channels and calcium ions are also not able to move through these channels, lead to an hyperpolarization state. This is very, very important to note. Okay, so this hyperpolarization state, once this is hyperpolarization state, this is around minus 110 millivolt. So this will be your membrane potential. So this is small mv. So this is your membrane potential over your rod cells when this is hyperpolarized. So it will lead to the decreased release of synaptic transmitters, so that your signals, means the light signal will not be decreased release of synaptic transmitters, so that the signal transduction can happen within your rod cells, so that the light signal can be read. So, okay. I hope this complete vision mechanism, how our rod cells in our retina are helping us to visualize in light and in dark, how they are regulated, is being clear, because yesterday and today, both the day, actually, I have discussed this thing with you guys. Now, before closing today's class, one important section which I have missed actually. Okay, so I told you that some of the kinases are specific for some of the amino acids. For example, generally we know which of the amino acids are being phosphorylated. We know serine, threonine, and tyrosine are phosphorylated in animals, and histidine and aspartate in case of prokaryotic cells or in bacteria. So 5 categories of kinases can be identified. One is able to phosphorylate both serine and threonine. So these are called serine threonine kinases. एक कैटेगरी है जो केवल टायरोसिन को फॉस्फोराइलेट करती है, सो दीज आर कॉल्ड टायरोसिन काइनेजेस. एक है जो ड्यूल स्पेसिफिसिटी काइनेजेस हैं, यानी किसी को भी फॉस्फोराइलेट कर सकते हैं, आइदर सीरीन, थ्रीरीन और टायरोसिन. Another category is identified which is able to phosphorylate the histidine in prokaryotes and aspartate. Okay, so there are 5 families which are able to phosphorylate the specific amino acids. Another thing I want to tell you in this particular class is, similarly, there are, because we know the function is opposite, kinases are opposed by phosphatases. So phosphatases are having 4 families. Serine, threonine specific phosphatases, which can remove the phosphate from these amino acids. Phosphatyrene phosphatases. There is another family found, this is very important again, which are either threonine or tyrosine specific phosphatases. Means they are not going to remove the phosphate from serine residues. Okay, and another family is which are in prokaryotic, phosphohistidine and phosphoaspartate. Means from this histidine and this aspartate are going to remove these phosphate groups from aspartate or histidine. So these are in prokaryotes only. So okay. Now there is 4 family of enzymes. One is called phosphatase, one is called phosphodiesterase, one is called kinase, and one is called phosphorylase. So these 2 enzymes are very close, and this kinase are opposed by phosphatases. These are having opposite functions, and there is one more category that is called phosphodiesterase enzyme, which break the phosphodiester bond and frequently required in signal transduction to degrade the cyclic AMP and cyclic GMP. Okay, so the phosphatases is an enzyme that remove the phosphate group from its substrate by hydrolyzing phosphoric acid with the help of some free hydroxyl group or water molecule. So they are able to remove the phosphate group. Now kinases, they are able to introduce a phosphate group from the ATP to a substrate, because they are using ATP here, and the gamma phosphate is always transferred to a substrate. They are called phosphotransferases. Similarly, the phosphorylase is an enzyme which do not require ATP, but they are able to add inorganic phosphate to an acceptor or a substrate molecule. So this is the basic difference. They are using ATP, and the gamma phosphate is added over substrate, whereas the phosphorylases do not require an ATP, but they are able to add the inorganic phosphate over this particular receptor or substrate molecule. Okay, so you need to remember these all four enzymes. They are very important in signal transduction mechanisms. Also, one more topic we are going to discuss is enzymology or advanced biocatalysis after we finish this particular signal transduction classes. Okay, so for today's class, this is what we have discussed is GPCR. Okay, so these GPCRs can either controlled via protein kinase A, यानी कि जैसा epinephrine signaling में होता है या adrenaline signaling में होता है. So it is able to phosphorylate nuclear receptors, for example, CRAB protein, which is a transcription factor. It is also able to phosphorylate some of the cytosolic proteins, for example, glycogen synthase, glycogen phosphorylase, and so on. Similarly, the one of the GPCR, for example, this rhodopsin is able to interact some protein, G protein that is called transducin. So this is important in our eye, with the help of which we are able to visualize things. So it is having important roles. So for today's class, this is all about today. So further we will move about the regulation or adaptation of these GPCR proteins in our next class. So the regulation of ion channels by G proteins. So actually this is something has been discussed, something we will discuss in our next class. And GPCR adaptation means desensitization, internalization, and okay, so these two things can happen. Either these receptor will be desensitized so that more and more ligand will bind and they will not respond. Either they are internalized, mean they will move inside the cell via some means some internalization structures will form and they will degrade it into the lysosomes. Okay, so thus they can be removed from the cell surface so that if actually happening in case of insulin receptors, they will be internalized. So, okay, and they will only come over the cell surface when there is insulin. Okay. So, okay, if you are having query now, this is the session for queries because we are running out of time now.